Welcome or welcome back to the channel. This is the second in my series of videos on chromosomes. Last time we had a brief primer on what chromosomes are, and then we discussed sex determination in ants, birds and mammals, chromosomal conditions in humans, and spooky clone bananas. This time we're going to talk about conditions like Down syndrome and what causes them, how different cells in your body become specialised to do different things, and we'll have a look at epigenetics, which means heritable changes which don't involve a change to an organism's genetic code, which is way cooler than it sounds, honest. As with last time, timestamps and sources down below in the description, and I'll put important definitions on the screen right here. So, we left off in the last video after talking a bit about Klinefelter syndrome. To recap, Klinefelter syndrome is characterised by a male phenotype but with two X chromosomes, XXY. This can cause symptoms like breast growth or decreased fertility. We also talked about X-inactivation, which is why individuals can survive with only one X chromosome, because they only need the products of one X chromosome. On a related note, Turner syndrome occurs when a single X chromosome and no Y chromosome is present. Symptoms include short stature, fertility issues, and webbed neck, among others. Now, when I started talking about symptoms of Klinefelter's and Turner syndrome, I might have raised a few eyebrows. If so, well spotted. How can there be any symptoms associated with these conditions at all? Don't all but one of the X chromosomes in a cell get inactivated? Surely an individual with Klinefelter syndrome would just have the extra chromosome inactivated, and an individual with Turner syndrome would just develop as if they had an X chromosome inactivated in each cell. Well, it turns out that X inactivation is a bit of a misnomer. Most of the genes on the inactive X chromosome are silenced, yes, but at least 15 to 20% of the X chromosome's genes escape silencing to some extent. This would account for the symptoms seen in these two conditions due to the idea of a gene dosage imbalance we discussed in the last video. A person with Turner syndrome would have a lower gene dosage of the X chromosome genes than is expected for an XX individual, and conversely a person with Klinefelter syndrome would have a higher gene dosage of the X chromosome genes than would be expected for an XY individual. With all this in mind, and with everything we discussed about human sex determination in part 1, I'll come back to my opening statement on the subject. We do tend to think that when it comes to sex and gender, sex is the less complicated of the two. Well, I don't know about less complicated, but as we can clearly see, it's not quite as simple as XX and XY being the final word on sex determination. Turner syndrome and Klinefelter syndrome are aneuploidies, a word which refers to conditions that come about due to an abnormal number of chromosomes being present. Down syndrome is a specific type of aneuploidy called a trisomy, which means that three copies of a chromosome are present instead of the usual two. In the case of Down syndrome, there are 3 copies of chromosome 21. Now, a trisomy can occur with any of the chromosomes, but apart from the X and Y chromosomes, the only trisomy conditions that can regularly occur involve chromosome 13, which results in Patau syndrome, chromosome 18, Edwards syndrome, and chromosome 21, Down syndrome. When other trisomies occur, they tend to be a bit more deadly. Looking at the chromosomes might give us a clue as to why this is. As you can see, the survivable trisomies seem to occur with the chromosomes that are a bit on the smaller side. And this does have something to do with it. However, chromosome 22, one of the smaller chromosomes, rarely has a survivable trisomy condition. The answer isn't directly to do with the size of the chromosomes. We talked about gene dosage earlier. For some genes, the gene dosage has to be pretty precise. Much more or much less of that gene being expressed is pretty bad news for the organism. Others, however, it doesn't matter so much. Chromosome 22 has at least one of those genes that require a more exact dosage, and due to the higher number of genes, it's more likely that a larger chromosome would have one or more of these genes. Trisomies occur to an event that happens before conception known as nondisjunction. Nondisjunction happens during meiosis, which is a type of cell division which creates gametes, sex cells. Unlike mitosis, which is ordinary cell division and results in 2 identical diploid cells, meiosis results in 4 haploid cells, which are statistically likely to be different from each other. During meiosis, the chromosomes are randomly reassorted, so with the 23 pairs of chromosomes, the total number of combinations is 2 to the power of 23, or 8,380,608 for a single sperm or egg cell. So you know, quite a few. And that's not even considering recombination, which is a topic for a bit later. Nondisjunction occurs in the first stage of meiosis when a chromosome pair fails to segregate, leaving one of the new cells lacking that chromosome and the other with an extra copy of that chromosome. Using our earlier aneuploidy examples with X chromosomes, the cell lacking the chromosome could lead to a gamete that becomes an individual with Turner syndrome, and the cell with the extra chromosome could lead to a gamete that could become an individual with Klinefelter syndrome. During the first stage of meiosis, homologous chromosomes line up next to each other on the centre line of the cell. Homologous in this sense means the chromosome 1s are next to each other, the chromosome 2s are next to each other, chromosome X is next to either another chromosome X or a chromosome Y, and so on and so forth. What allows this to happen is a physical connection between the arms of each of the chromosomes, sites where they cross over. This connection is maintained by something called the Cohesin complex. A complex in this sense refers to multiple protein subunits working together as a single unit. The Cohesin complex forms a ring around the two chromosome arms to maintain this connection. This connection allows the chromosomes to connect to the spindle fibre, which is a structure that physically pulls the chromosomes to opposite ends of the cell. The connection gives some resistance against the pull of the fibre so that no chromosomes are separated too early, and everyone gets lined up properly. When it's time for the homologous chromosomes to be pulled to opposite ends of the cell, one of the cohesin subunits is cleaved by an enzyme, conveniently named separase, and the ring breaks and the chromosomes separate. Nondisjunction that occurs at this stage of meiosis is caused by the separation happening too soon. This can happen due to a variety of reasons, but interestingly, one of those reasons is the physical placement of a crossing over site on the chromosome. When the site is closer to the end of a chromosome arm, the tendency for loss of cohesion increases. This seems to be because the cohesin ring can slide off the ends of chromosome arms, and the further up the cohesion site is, the more likely it is to happen. Before we move on, I should mention recombination. At this stage of meiosis, there's a small chance that the little bits of chromosome that are past the crossing over point can swap over. This uses the same mechanisms as those used to fix double-stranded breaks in DNA. This means that the number of combinations of different chromosomes in a gamete I mentioned earlier, 8,388,608, can just be thrown out of the window entirely. The real number is way bigger than that when you account for the fact that chromosomes can just swap bits and pieces of themselves as they please. You might have heard that the chance of conditions like Down syndrome occurring increases the older the mother is at conception, a phenomenon known as the maternal age effect. Most aneuploidies in eggs from older mothers arise in phase 1 of meiosis, the phase we've just been discussing. Oocytes, or eggs, develop up to this phase and then stop there until ovulation, so all the eggs are at this stage until birth. This has led to the theory that the maternal age effect is caused by an increased chance that the crossing over process will fail and the paired chromosomes will separate prematurely the longer it goes on. This theory is supported by a 2005 study that used a mutant line of mice deficient in a gene for a cohesin subunit specific to meiosis. The chromosomes of the mice's eggs were observed at different ages, from 1 to 12 months old, and the occurrence of nondisjunction increased massively as the mice got older, suggesting that the loss of cohesion is directly related to nondisjunction and therefore aneuploidy. We've talked a fair bit about genes and gene dosage, so I think it's time to have a quick and rough overview of what genes actually do. Genes are sequences of DNA nucleotides that encode proteins. Nucleotide is just the name of the molecules involved. The nucleotides are called cytosine, guanine, thymine, and adenine. The names aren't important for this part, so just think of the letters C, G, A, and T. You might know that DNA is double-stranded. The nucleotides on one strand bind to those on the other strand, C to G, A to T, and we call these complementary pairs. On the way from DNA to protein, we have another step, RNA, which is another type of nucleic acid similar to DNA in ways we don't really need to go into much detail in. We're not doing full-on biochemistry here, thankfully. RNA nucleotides have the same names as their DNA counterparts, except we have a molecule called uracil instead of thymine. The gene sequence of the DNA undergoes a process called transcription to create a molecule of messenger RNA, essentially a copy of the gene's complementary pairs, but as RNA instead. The last step is translation, which is when the messenger RNA is used essentially as a bit of code to assemble a protein from amino acids. I'd love to go off on a tangent on how that works, but I'm going to try and stay on track for now and leave you with vague assurances that I'll cover that in another video. I've mentioned differentiation a couple of times in this video and in the last one, and now it's finally time to talk about it. Differentiation is the process by which cells in a multicellular organism, like you presumably, become specialised for their role. For example, erythrocytes, red blood cells, being able to carry oxygen. The different types of cells all have the same genetic material but produce different proteins in different quantities to perform these different tasks. This is why your red blood cells aren't hairy and your skin isn't red with haemoglobin. The way this happens ties into what we were discussing before about Klinefelter syndrome, epigenetic gene silencing. Epigenetics, as I said near the start of the video, is a term that refers to heritable factors determined by things outside the genome, so not changes to the actual code, if you will, but rather changes to things around the code that alter how the code is read. As an example, the Professor Conrad Waddington, who coined the term epigenetics, was working with Drosophila, fruit flies, because of course he was. You can hardly do anything in genetics without involving fruit flies. Anyway, all Waddington did was incubate the Drosophila eggs at a slightly higher temperature than usual, and when they had fully developed into adults, they had a pair of legs where the eye should have been. Nothing about the fly's genome had been changed, but rather a previously silenced gene had become unsilenced. He called this gene Aristopedia. Aristopedia is what is known as a homeotic gene, which is a gene that regulates the development of body parts, like legs. Homeotic genes usually encode transcription factors, which lead to more genes being expressed in a sort of cascade, and lots of different things happening as a result of that which wouldn't have happened without the first step. We saw something similar in the last video when we talked about testosterone, leading to a cascade of developmental events further down the line within the same principle. What Waddington saw was essentially a gene for a leg being expressed in a part of the body where it ordinarily wouldn't be. I mentioned before that small chemical groups can be added to DNA and be passed down that cell's lineage. So if I added a methyl group to a particular bit of DNA, then the two cells that cell divides into will have a methyl group on that same bit of DNA. This is because of an enzyme called DNMT1, which is a DNA methyltransferase. Belter name for an enzyme that. The name tells you exactly what it does. It puts a methyl group, transfers it, onto a piece of DNA. When it happens upon a piece of DNA where only one side is methylated, it'll add a methyl group to the other side. And then when the cell is going to divide, the DNA will be replicated, each strand will go off to form a new double strand of DNA, and the new strand will get methylated by our mate DNMT1, and there you go. Heritable changes to your DNA that don't involve changing the code. Epigenetics. I mentioned DNA methylation because it's one of the ways epigenetics are used during differentiation to downregulate or silence genes in different cell lineages. Transcription factors can find it harder to bind to areas of high DNA methylation, so any gene in that region of DNA is either much less active or might as well not be there. There are other modifications that can be made to DNA such as acetylation, hydroxymethylation, and others that lead to a whole landscape of potential differences between cells containing the same genetic information with wildly different patterns of gene expression. So that this process can begin again for an organism's offspring, these epigenetic markers are pretty much reset when new gametes are generated or when a new embryo is formed. This is also quite helpful for the health of the offspring too, as we can actually pick up some of these epigenetic modifications during our lives. You're probably already aware that smoking could cause DNA damage in lung tissues, but it's found to also cause DNA methylation, histone acetylation, and other epigenetic modifications, with the broad effect that the expression of genes for inflammatory proteins is altered. There is evidence that some epigenetic changes manage to avoid the reprogramming, however. For example, the children of fathers with pre-diabetes or diabetes have been found to be more susceptible to diabetes themselves. This alone isn't grounds to suspect epigenetic inheritance, but it's made more likely by the presence of similar DNA methylation patterns in both the fathers and the children, with these particular patterns being associated with insulin resistance, which is a key factor of diabetes and pre-diabetes. So it seems that despite 2 rounds of removal of epigenetic markers, some manage to escape and get passed on to the next generation. Remember Charles Darwin? Probably, right? His theory of natural selection is our basis for how we think about evolution. Organisms are born with a set of characteristics, and there's variation of these characteristics in the population. Parents pass down their traits to their offspring, and individuals with traits that suit the environment survive, for long enough to pass on their traits anyway. The next generation is then more likely to have these favourable traits and survive, and so on and so forth. You may also remember being told about this other guy, Jean-Baptiste Lamarck, who put forward another theory. Organisms develop traits over their lifetime and similarly pass on those traits to their offspring. The famous example being the giraffe, which supposedly started out as something resembling a big off deer with crap camouflage and wearing dealy boppers, which would stretch its neck to reach high leaves on trees. Over time, supposedly, its anatomy would slightly change, its neck would stretch, and its offspring would have longer necks too, and then they'd stretch too to reach higher leaves, and well, you get the picture. Clearly Lamarck was barking up the wrong tree with this, and even if he barked up the right tree, it still wouldn't stretch his neck and allow him to pass on that trait to future generations. I think you see where I'm going with this though. These epigenetic changes are acquired during a person's life, and then passed on to their offspring, just like Lamarck's giraffes. That's obviously not a reason to ditch Darwin and go full Lamarckian, but we can see that in certain situations where these modifications have escaped the epigenetic reset, there is an element of Lamarckian inheritance. Pretty wild. Also, I know I've been making a few jokes at Lamarck's expense, but to be fair to him, he was mostly a botanist, and plant epigenetics work a little bit differently to our own, with many more epigenetic markers being passed down to offspring to allow for adaptation to environmental pressures. Anyway, that's about all for today. Thank you so much for watching. Please consider subscribing to the channel and drop a like on the video if you fancy, and I hope to see you next time.